Okay, it seems that the screen is well on its way. Okay, let's start. So this is what I will present today. JuLI Stage, a new standard of cell history recorder. These are the contents for today's webinar. I will start with the latest trends in the instruments and publications, followed by CHR, stands for the Cell History Recorder. I'll talk on launching the new standards of cell history recorder, JuLI Stage. These are the trends in the journal publications. Since 2006, increasing number of researchers and scientists are submitting their results in video form to the journals. This graph shows the number of publications from a particular video-oriented journal called JoB, which stands for the Journal of Visualized Experiments. The video publications have increased of approximately 27 times from the starts. The reason for researchers and publishers to publish their valuable results with a video recording are to present concrete verification and confirmation of their time-taking data. In future, I hope that JuLI Stage as CHR could help in providing such precious results for faster publications with high impacts. Several researchers also have suggested that number of higher quality journals would like to see the video recording of the experiments and results. And recently, JoB also made an agreement with famous journal called Science. This slide shows a popular video data upload website, DNATube and JoB. DNATube contains approximately 23,000 experimental videos, and JoB has close to 5,000 video publications. Other journals, such as Nature Family journals, are requesting scientists to submit their video recordings of their data. Hence, increasing number of researchers and publishers are relying on the video recording in presenting, reviewing, and validating their data. This clearly shows a new trend in publication community. It is not an easy task for scientists to video record all their crucial experiments and results. It is not easy for any researcher or scientist in the past. Even now, tasks are more depending on experimental instruments and environments, especially the traditional visual fluorescence microscope or confocal microscope needs extensive training to be skillful, and the results are many times depending on the level of training of technicians and scientists. With the JuLI Stage, this task can be performed easily by utilizing various simple video recording functions within the instrument. We hope that we can lead the trend and contribute to the community by helping researchers to submit their valuable results to the high-impact journals. We'd like to present JuLI Stage as CHR. As I mentioned before, in order to submit your precious results to journals, you have to present both detailed changes in cells or tissues. Often, high-throughput cell-based assays would require additional supporting data, such as automatic growth curves and endpoint analysis. CHR would do that as a default by taking the history of cells. Therefore, we decide to name JuLI Stage as a cell history recorder for a simple and direct concept. Why should researchers use CHR? As we all experience, that the traditional method relying only on endpoint experiments. Diverse limitation could occur, placing microscope into the incubator, and there could be many interruptions during the transportation of flask between incubator and microscope. In turn, this could affect the final result. You would have to wait until the termination of the experiments before verifying whether it was performed properly. If something went wrong during the experiment, we may not know until the end. With the cell history recorder, we can change this. We can record the events of cells from the beginning until the end. We can observe the cell in real time, and we can also go back to the exact time point whenever we want for optimal and best results. In a sense, we can hear and see what cells are telling us in real time. CHR will definitely value up your research, and most of all, it will save your time. CHR, in a sense, is a new concept. This slide shows a current method of CHR, monitoring cells which cycles between experimental observation, incubation, and the data analysis. The traditional analysis could take very long time, and many times it could be prone to frequent human errors. Especially low quality data could be obtained from a 96 or 384 well-based high content screenings. This slide shows the current method of automated CHR, which was built around the microscope. It created a cell incubation environment around the microscope, which demands high cost. It could be complex and unstable. And many times, the results are depending on the levels of a skilled researchers. I'd like to introduce JuLI Stage as a new standard of CHR. We developed JuLI Stage to save time, energy, and resources for researchers and for the cost for institutions. So the researchers could concentrate your time and energy on the experiment. JuLI Stage is 3-in-1 real-time cell history recorder. JuLI Stage could take images and video recordings of cells and their changes. Without additional tools, JuLI Stage can analyze your results. We believe that the JuLI Stage will open doors in CHR concept. Pictures in the left shows a typical microscope-oriented cell monitoring system. JuLI Stage on the right side, it is a all-in-one instrument, which can be placed inside of your incubator and record experiments, whether it's inside or you can take it out and record it from the outside. Hence, the cell-based experiments can be proceed in a stable condition because researchers do not have to take the cells out from the incubator for observation. The experiments can proceed in an incubator as long as you want to observe cells without any problems or interruptions. In a sense, you can create a very stable experimental environment for your valuable results. JuLI Stage can fit into small space, and you do not need additional equipment purchase or extra cost. JuLI Stage will let you take only 3 steps to create a very simple image and simple video recordings. Unlike conventional complex CHR, JuLI Stage can let you perform many cell-based experiments easily and quickly with just few clicks. In the past, when you want to obtain results from 96 separate experiments, it could take up to maybe 1 or 2 months. But now with the JuLI Stage, it will only take maybe 2 days in parallel setting. Now I'd like to mention several functions within JuLI Stage. This is a multi-well monitoring function. You can monitor up to 384 wells even simultaneously. JuLI Stage has a multi-positioning monitoring function, as you can see on the red dot. Wherever the position is, you can monitor as many as you want simultaneously. As you already know, image revision could take very long time even after experiment is finished. The traditional system needs separate editing program. Powerful software program could support researchers and make their job very easy for the analysis. Researchers could save time and take a break and perform or plan for other experiments. For that same reason, we believe that JuLI Stage can provide this, and it can be an answer to above issues of analyzing image and editing videos. JuLI Stage provides easy and powerful editing function built in within the instrument. Overlaying image, video cropping, frame rate control, image stitching, deep phase, contrast, all these functions are available within JuLI Stage. Let me show you one of the editing function. After performing a fluorescence experiments, you frequently use image merge function, right? JuLI Stage has image merging tool called overlay. As you can see on the left, you can merge bright field image, green fluorescence, red fluorescence, dark field fluorescence images all into one, as you can see on the upper right-hand corner. And this slide shows image stitching function. Stitching is a process of combining multiple photographic images with overlaying field of view in producing a segmented panorama or high resolution image. And this is an example of our stitching image. As you can see, there's no trace. Isn't it great? This slide shows our image after cropping. On the left, we are cropping from the left side to the right side. In the printing, cropping refers to a removing unwanted area from a photographic image. In this software, you can do this very easily by one click, drag, and to make a crop image. And this slide shows one of the powerful editing functions called frame rate control, as I mentioned before. This function will allow you to control the video play speed as you intended by using the frame rate control function. And the left side shows a slow frame rate control and the faster on the right side. And this slide shows the dephase contrast function. After obtaining a blur image, which is very difficult to see on the left side, with the dephase contrast tool, you can have a sharper and the better results. And there are numerous applications for JuLI. There are numerous applications for JuLI Stage. As I mentioned before, you can carry out experiments in a stable conditions, acquire image, video record all the changes, and perform data editing easily. All these things are available within the JuLI Stage. As I listed here, JuLI Stage can perform various applications from the cell proliferation, wound healing, apoptosis, angiogenesis, 3D spheroid experiments. And now I'd like to show some of the data we obtained by using the JuLI Stage. This slide shows cell proliferation data recorded by JuLI Stage. We combined the green fluorescence and red fluorescence expressing same USO2 stable cell line. And cell growth images were captured for over 72 hours with interval of 15 minutes. And this slide revealed the results from a cells monitoring stem cell culture. Mesenchymal stem cells were transfected with a targeted DNA. After the induction, neuronal differentiation and cell growth images were captured for over 144 hours. This experiment was performed in collaboration with the Cha Hospital. As you can see, the cells are growing from the middle to the outward, and you can see that they are forming edges, and the neurons are differentiating, extending. You can see some of the pseudopodias. This slide, again, clearly shows one of the most exciting cell differentiation and division data. Stem cell monitoring were performed for 72 hours with the mouse embryonic stem cells. As you can see, the cells are dividing from one cell to two cell stage. Now we can see the full cell stage. From the full cell, it will divide into a cell, into 16 cell stage, and it will divide into 32 and 64. And after 64, it will form a blastula, and you can clearly see the blastocore inside. And we stopped the experiment before the gastrulation. As you can see, we can see clearly the blastocore, and cells are continuously dividing and expanding and growing. We can all capture all this within JuLI stage. This slide shows one of the very simple and very straightforward assay called wound healing assay. A scratch was made with CHSY5Y cell culture, and it was incubated for 50 hours. And let's look at the video, and you can see that cells are growing and filling the gaps, and there are, we can see that cells are, very clear, and there's no wobbling. They're very stable, and they're filling the gaps, expanding and growing. And this another experiment is very exciting. With this experiment is called 3D spheroid assay, that we again made a spheroid with green fluorescence and red fluorescence expressing U2OS osteosarcoma cells. We co-culture them and monitor for 10 days. As you can see, the cells conform to the middle, to the center at first, and then now they are expanding and growing towards the outside. And many cancer researchers are studying this various drug interactions and cell-cell interactions using this 3D spheroid way of looking and observing the cells. And this reveals another the cell growth, very simple experiment. This was performed in collaboration with the Semmelweis University. This is a time-lapse image of fluorescent intensity in osteoclast cells, where it was recorded for 3 hours with 10 to 20-minute intervals. Left side is a control, and right side we can see the fluorescence after transfection with the EGF treatment. And next slide, this is another exciting experiment. Is cytotoxic experiment was carried out in collaboration with the Poland Warsaw University. Left side shows a control of a neuron cell monitoring, and the right side shows a neuron cells after infecting with the herpes virus, EHV1. As you can see that this herpes virus are destroying and killing neurons, and the cells are being uplifted from the bottom of the flask into the solution. And you can see various debris, where on the left side, neurons are growing very stable condition. Okay, this slide also shows one of the very prominent experiment called apoptosis, that we saw the effective camptothecin on U2OS red fluorescence stable cell line. We recorded for 47 hours with the 20-minute intervals. That with the time-lapse JuLI stage images, we added caspase 3-7 reagents. After apoptosis occurs, released and activated caspases can be monitored with green detection reagent, as you can see on the screen. The cells are dying and releasing caspases into the solution and interacting with the caspase substrates, producing green fluorescence. And this slide shows the transfection efficiency experiment. After PA317 cells were transfected with red fluorescence gene, cells were monitored and quantified for their transfection efficiency for 40 days with an interval of 20 minutes. That cells took the red fluorescence gene are being expressing this red fluorescence protein, and we can observe this with the JuLI stage, and which we can know which cells the uptake the red fluorescence gene. And now this shows the specification of JuLI stage that we have a light source for the light source we have blue, green, and UV LED. For objective lenses we have 4x, 10x, and 20x plus the digital zoom capacity. For fluorescence, we can monitor up to 3 fluorescence: DARP, green fluorescence, and red fluorescence. For camera, we have a high sensitivity monochrome CCD camera from Sony. So there are other specifications you can see on the screen. Thank you for listening, and your thanks for participation to today's webinar. And that's all, folks. And now I'd like to end today's webinar with a short movie made for JuLI Stage.
JuLI Stage, the real-time cell history recorder. Compact, compatible live cell imaging system from inside your incubator.